Hello and welcome. My name is Jan Detmers. I'm the VP of Single Cell and Spatial Phenotyping at a company called Doppelgänger Biosystems. We are a CRO doing sample analysis for preclinical and also for clinical trials, but we also support discovery projects. Today I would like to talk about metabolic phenotyping. My talk is called Seahorse analysis for tissue. And in the beginning, I would like to give a quick overview about the next 20 minutes. I would like to differentiate between metabolomics and metabolic phenotyping. I would like to explain a little bit the Seahorse technology in contrast to QSM metabolic phenotyping, which is an alternative system to the Agilent Seahorse platform. And I would like to speak today about application in tissue analysis. And at the end of the talk, I have two case studies. One is on oncology, and the other one is on immunology. First of all, metabolomics and metabolic phenotyping, this is often confused. Metabolomics is the science where basically through often mass spec-based platforms, we quantify a concentration of a given metabolite, let's say a fatty acid. In metabolic phenotyping, we measure rates. And there are two platforms in the market. One is the Seahorse system by Agilent, and the other one is the QSM system by Doppelgänger. And what these platforms do is they determine a rate. So it's a metabolite turnover rate. In case of Seahorse, for example, there are two parameters. One is the OXPHOS rate, and the other one is the oxygen consumption rate. So Seahorse needs cell suspensions, and these cells also need to be happy. And then you can determine the basal rates, the maximum rates, and then you can calculate the metabolic reserve. In contrast to that, QSM is not only compatible to cell suspensions, but also to tissue. This is what I would like to talk about today. And this includes also FFPE tissue. This is very important for clinical trial support. But we can also work on single cell metabolic phenotyping, and we run the first spatial metabolic phenotyping projects now. QSM, in contrast, measures again the basal level, the maximum level, and also the metabolic reserve. But we typically work with 8 parameters: the ATP production rate, obviously one of the key metabolic rates. You want to look at cell viability and also toxicology related effects. Very closely linked to that is the oxygen consumption rate. We also look at the ATP oxygen ratio, because this gives us a very good overview on the efficiency of the electron transport chain and the oxidative phosphorylation in total. Going deeper into why a cell switches, for example, from fatty acids to a glucose metabolism, we look at the substrates, mainly glucose uptake rates, lactate uptake rates. Often these rates are negative because cells are exporting lactate. We look at fatty acid uptake rates, but we also look at branched chain amino acid uptake rates and ketone body uptake rates. And initially, we have validated our platform against the Seahorse, because obviously this was the gold standard in the market. Here below, you see an experiment where a cell line was drugged with 3 different compounds, and you see that the results are basically identical. The QSM is available in 9 flavors. Our most important product is the OXPHOS metabolism, but we also look at liver metabolism. We have a few additional parameters here where we look at beta oxidation, for example. The Triomics kit focuses on the integration of metabolic phenotyping data with transcriptomic and with proteomic data. Cardiometabolism obviously is used in cardiology. Neurometabolism is very interesting. This is a product which focuses obviously on CNS diseases, and there obviously the energy metabolism is of utmost importance. There we have the neurometabolism kit. Safety metabolism addresses toxicology relevant questions. Our main focus is on DLE. Single cell metabolism is the product for single cell metabolic phenotyping, very similar to immunometabolism, where we particularly focus on the metabolism of immune cells. Cancer metabolism focuses a little bit on both. So we look at the metabolism of the cancer cells, but also the metabolism of the immune cells in the tumor microenvironment. And the platform is validated for human, for rodent samples, monkey, and for rabbit samples right now. So to give you a quick overview on the metabolic phenotyping features, here on the left side, we look at ATP production rates, and we look at CAR T cells that have been sorted from a PDX mouse model. On the right side in red, you see the wild type CAR T cell, and on the left side in blue, you see the an enhanced CAR T construct, and both CAR Ts have been applied to a mouse model, and then the CAR T cells have been harvested from the PDX mouse model, and you see that the ATP production rate, which is very important if you think about exhaustion processes, that this is about 25% higher as compared to the wild type construct. On the right side, you see T lymphocyte subpopulations. Again, we look at ATP production rates, always determined in gram per hour. And here you see that the ATP production rate of T lymphocytes varies by almost 100%, right? So depending on the T cell subpopulation, we measure very different ATP production rates, also very different oxygen consumption rates, and this can be monitored with this platform as well. These lymphocytes have been sorted from a liver cancer patient. And then we look at the spatial metabolism. Here we worked with glioma samples. We used the nanostring/Bruker geomics platform, and we measured ATP production rates, oxygen consumption rates in the middle, and glucose uptake rates. And we measured kind of a transect. So the blue bar is the healthy adjacent tissue. Then we measured the periphery infiltrating parts of the tissue, and then the core in purple, the core of the glioma. And moving from outside to inside the tumor, you see that the ATP production rate slightly decreases. Same with the oxygen consumption rate. It slightly decreases. At the same time, cells turn glycolytic, and you see an increase of glucose uptake rates. And we will have more applications in the very, very near future. So most of our customers use the QSM process either for translational purposes, like you see here on the left side in this NASH project. You will find these posters also on our website. In the middle, this is a Parkinson screening model. This was kind of a model validation project, and we've pretty much focused on the energy metabolism in that project. And the colitis, again, is a translational project where the sponsor ran a mouse model. The question was, how much is the mouse model really reflecting the situation in chronic colitis patients. The first case study I would like to highlight today is an oncology study. And here we analyzed tissue biopsies from low-grade glioma patients. And these patients have a rather rare mutation, IDH1-2. It's a gain of function mutation. So basically, in the citrate cycle, these patients produce a byproduct which has two effects. First of all, the energy metabolism loses quite a bit of energy in the metabolism, and obviously the byproduct enriches in the patient brain. This was a phase 1 study including 49 subjects. And the sponsor here compared two compounds, vorasidenib versus ivosidenib. And the mode of action of these two compounds is slightly different, but it basically blocks this gain of function mutation on the enzyme side. So basically, this bypass is blocked, and through this, the citrate cycle works as expected again. And the purpose of the study was really to evaluate whether the energy metabolism in brain tissue samples can be used as a pharmacodynamic marker. And the purpose for the sponsor was really to get an idea about the efficacy of the drug and ultimately to take a decision which compound to move into phase 3. We measured the ATP production rate and we looked at the oxygen consumption rate, and also we monitored the substrate utilization, and you will see we can see a shift here. So these patients received both compounds in a randomized study, upfront surgery, and when the therapy was successful, finally the patient underwent surgery to remove this low-grade glioma tissue from the patient brain. On the left side, you see the ATP production rate, and you see that through the action of vorasidenib, here we don't have a negative control, right? We basically compare the efficacy of two compounds. And we see that obviously vorasidenib stopped this gain of function mutation in a more efficient way, and you see an increase of ATP production rate on average by 28%. So this is really significant. Please keep in mind these are brain samples, and typically the brain on the metabolic side runs really very constant. It's a very protected environment, and typically these cells do not undergo dramatic changes. But 28% increase in ATP production rate, this is a very dramatic increase. And we validated these findings by the oxygen consumption rate, and this is in the same order of magnitude. In fact, it's basically almost the same, an increase in 27% in terms of oxygen consumption rate. And now the underlying question is there a change in the substrate utilization? And you see that primarily these compounds seem to affect the substrate utilization, and you really see that this increase in ATP production is a result of an increase of fatty acid utilization. So here we can see that QSM can effectively monitor a treatment-induced shift in substrate utilization in the patient. And here we can use ATP production rate and oxygen consumption rate as efficacy biomarkers, basically as PK biomarkers that have the potential to be used as a clinical endpoint in future glioma trials. And by the way, vorasidenib, the compound which showed a significantly better efficacy here, was advanced to phase 3 testing. In the meantime, The drug has been approved and is sold under the brand name Vorangio. The second study I would like to highlight today is on the immunology side. Again, this is a clinical trial, and we work with samples from psoriatic skin, and these are punch biopsies from the skin from the same patient before and after treatment. The compound was secukinumab, a monoclonal antibody that was used on this phase 2 clinical trials. Here we look at a broader picture in terms of metabolic phenotyping. And again, the idea was, can we use these parameters really as a PK biomarker? So on the left side, you always see the parameter pre-treatment, on the right side, post-treatment. And we start with the ATP production rate. And you see that secukinumab decreased ATP production rate by 45%, which is really significant. And again, to validate this, we look at the oxygen consumption rate, and it's in the same order of magnitude. So basically, inflammation takes a lot of energy. The energy requirement of inflammatory constant and chronic inflammation processes is really significant. And through the secukinumab treatment, we can show that this really returns to normal. And we see the same for the glucose uptake rate. It's basically the same reduction we see for the oxygen consumption rate. The glucose uptake rate decreases, and the same is true for the fatty acid uptake rate. And what we can really see here is that an inflamed tissue returns back to normal. We can validate this by multiple parameters, and this aligns with the clinical improvement. Happy to send you the publication if you want. And I think here we can really see that the cell energy can really serve as a biomarker, and QSM is ready for clinical trial support. So in summary, I would like to highlight, please don't confuse metabolic phenotyping, where we look at rates, very functional parameter, versus metabolomics, where we look at concentrations, which is important, for example, if you want to monitor storage effects, let's say in fatty liver disease, for example. And there are two main platforms. One is the Seahorse platform. It's a very popular instrument, which is often used in discovery settings. And there's QSM, metabolic phenotyping. QSM, by the way, stands for quantitative system metabolism. And this is compatible also to tissue phenotyping, to single cell phenotyping, and also to spatial metabolic phenotyping. We looked at two case studies today, and please keep in mind, this is really an application where there is no other equipment in the market right now, and we offer this as a service. So in the chat now, I can see a question. Can you talk about the technology? Yeah. I should focus a little bit more on the QSM technology. So basically what we do, people send us cells, tissue biopsies, FFPE biopsies, organoids, you name it. Serum plasma are not compatible in this platform. And if we have enough sample, we do enzyme proteomics. We specifically look at 518 enzymes, and we determine the amount of these 518 enzymes. If we don't have enough material, we do an enzyme transcriptomics approach, where we quantify expression levels of mRNA. And this goes into our omics data platform. And the platform has basically, and this is hard-coded, the central metabolism is catalyzed by these 518 enzymes, including their isoforms. We know the regulation of each enzymatic reaction. Most of these enzymes have been crystallographically elucidated, so we know the structure, and most important, we know the enzyme constant. And then the only missing variable is the amount of enzyme present in a given tissue or in a sample. And this is what we get from the enzyme proteomics assay. And then the platform calculates the metabolic rates within these 22 central metabolic pathways, and we report the data in these 8 parameters that I mentioned early in my talk. A second question is more on the, so the validation. We used Seahorse really as the gold standard, meaning that we run a bunch of assays in Seahorse and parallel to the QSM platform. And the results are basically the same. The absolute quantification is not identical, but the relative quantification is very, very similar. And then on the right side, you can see that we used also biochemical assays to validate as many of these parameters as possible. The coverage of the platform covers 22 pathways, and depending on what the application is, we report between 8 and 22 parameters. Right, and with this, I would like to thank you for today. If you want to download the posters, please go to our website, doppelganger-bio.com, or contact us at contact@doppelganger.com. I have more webinars in the near future. One is on single cell metabolic phenotyping, where we can really nicely look at T lymphocytes subpopulations in a tumor, and we can measure basal rates, we can measure max rates, and we can calculate the metabolic reserve. And we look at T cell subpopulations in terms of their ability to produce ATP, and also to look at exhaustion effects. And below you see one of the first projects that we ran on spatial metabolism, where we used the 10x Visium platform. And with this, I want to thank you, and please tune in for one of our next webinars. Thank you, and have a nice day.